What are enzymes? Enzymes are catalysts that speed up the rate of reaction without being changed or used up in the reaction. It means that catalyst is only to speed up the reaction. It is not being changed or used up in the chemical reaction. A group of protein is known as enzymes. Biological catalyst. It means enzymes are called as biological catalyst. Biological because they are made in living cells. Catalyst because they speed up the reaction without being changed. Necessary to all living organisms as they maintain reaction speeds of all metabolic reactions, all the reactions that keep an organism alive at a rate that can sustain life. Here, the metabolic reactions are breathing, digestion, and the circulation, etc. For example, if we did not produce digestive enzymes, it would take around 2 to 3 weeks to digest one meal. But if we use an enzyme, it takes around 4 hours. It doesn't mean that we use. It is basically natural in our human body. For suppose the saliva is the enzyme produced in our mouth. When we are chewing the food, the salivary glands produce enzymes known as saliva in which the speed of rate of reaction is increased. For an enzyme to act with substrate, it is necessary to the enzyme to be specific to the substrate. For suppose substrate is specific to enzyme 1. How can we determine it? Because we can see the complementary shape. Enzyme 1 is complementary in shape to the substrate will catalyze a reaction, while enzyme 2 is not able to bind to this substrate, they are not complementary. Here we are required to understand the lock and key hypothesis. Hypothesis means theory. Here the enzyme is the lock and the substrate is the key. Enzyme substrate complex. First of all, in the first stage, the substrate will come towards the active site of enzyme. Active site is the place in the enzyme where the substrate is attached. And the reaction begins, which break down the molecules into their simpler substances.